Okay, Chapter 5, Section 4, Steps in the Replication Cycle. Some of the differences we're going to see between prokaryote and eukaryotic viruses is not only going to be in the nucleic acid, the genes, but how the nucleic acid actually gets into the cell itself, the host cell. If it's a prokaryotic one, remember the virus, the capsid and all the tails and all that stuff actually stays outside. Whereas with the animal, well, it actually, for a lot of them, the whole virus gets in. The capsid, the nucleocapsid and all gets into the cytoplasm. And then the genome will be released. But whether you're talking, the bacteriophage, an animal, plant virus, we see that there are a certain number of steps that are always going to be in common. Attach, penetration, synthesis, assembly, release. Got to be touching, got to somehow get in, whether it's the entire virus gets in or you just inject the genome. The virus takes over and starts turning that host cell into a virus-making machine. The capsomeres form the capsids, which will then package around the genome. And then somehow those newly made viruses have to get out. Come into contact. So this is going to be a bacteria since the virion's staying outside. Remember, virion, a singular for virus. Virus actually is a plural. So a virion, singular, attach. Genome gets ejected in somehow, gains entry, takes over the host. Host stops all host cell metabolism, host cell, protein synthesis, host cell genome synthesis, because all the amino acids, all the nucleotides now are going to be utilized for the making of virus components. Make the components, assemble the components, virus has to get out, virus has to be released. We see that there is a pattern to this, where the bacteria, when they were growing, remember you had the growth curve, lag, log, or exponential stationary decline. Well, it turns out the replication cycle of a virus also has some phases to go with it. They're not going to have a growth curve, similar to the bacterial one, okay? Because theirs is more of a one step. Get in, make a lot, get out. Done. There's no stationary, nothing like that. So we see that there's different phases, the eclipse phase, the maturation phase, the latent period. The eclipse phase, virus gets in, and then you start to see different things being made. The early enzymes that are going to take over the cell. They're going to then trigger the nucleic acid, the copies of the genome being made. Then the protein coat, the capsomeres will start to be made. This is the eclipse. Students always ask, why do the nucleic acids have to be made before the capsomeres? Well, think about it. If only one copy of the genome is getting in, you can only read and make messenger RNA off of that copy so many times or so quickly. But if you take that genome and then you make many copies of it, each of these copies can now be transcribed from. So that's why you see the nucleic acid generally comes first. Then you get the protein coat. This is all the eclipse. Then you move into the maturation phase. You now have enough genome. You now have enough capsomeres forming capsid so that you can start assembly and release. Now, if this is a naked, it's going to just burst the cell and everybody gets released all at once. If it's an enveloped, as this maturation occurs, you're going to see these viruses exiting out slowly. So that way it can maintain the cell. Because if you have these all exit out too quickly, all of them tearing off pieces of the cytoplasmic membrane, it can only occur so long before the cytoplasmic membrane just completely breaks down and cell dies, whether the viruses are finished, assembly or not.